Hello, my name is Jory Van Cleve. Today we're going to be talking about glycolysis, glucogenesis, and the role of aldolase plays in gene expression. Glycolysis is a series of reactions that breaks the 6-carbon sugar molecule into two 3-carbon molecules. Glycolysis is the first stage of cellular respiration, which is how we get our energy from the food that we eat. Glycolysis is a pathway that can function with or without oxygen. Therefore, many different organisms can utilize this process. Glycolysis occurs in the cytoplasm of a cell that includes two main phases, the energy requiring phase and the energy releasing phase. During the first phase, glucose is taken in and receives two phosphate groups. As glucose turns to fructose when being rearranged, it is broken down into two 3-carbon sugars. This part of the phase uses up two of the ATP molecules. One of the paths results in DHAP or DHAP, while the other results in glycerol dehyde-3-phosphate. The second path can continue to the energy releasing phase. During this phase, the two 3-carbon sugars end up creating pyruvate. This also results in two ATP and an NADH molecule being produced. However, this part of the cycle occurs twice, therefore it ends up having double the amount of ATP and NADH. The enzymatic actions of glycolysis can also contribute to the growth of tumors, the cell cycle, and the spread of cancer. And so if you look on this graphic, it shows glycolysis, and it shows the metabolic reactions happening, shows the enzymes being a catalyst and breaking that glucose and producing that pyruvate. Glucogenesis is a pathway where glucose is created from pyruvate and lactic acid. It is regulated by two separate enzymes. I'm going to try to say these. Phosphoenolpyruvate carboxyl or PepsiK, and glucose 6-phosphatase, which is also called G6-pase. Glucogenesis is an important process needed to regulate blood glucose levels, especially in preventing low levels. Carbohydrates are not needed in the diet for this process to take place. It can take place even when someone is on a low-carb diet, if they're fasting, if they're malnourished, or just having a hardcore, vigorous exercise. When this happens, glucogenesis can continue with substrates that come from protein sources, lipids, glycerol, and other fatty acids. The vast amount of glucogenesis occurs in the liver, along with the aid of the kidney. So when blood levels need to be increased, glucose is dumped back into the bloodstream. Insulin then jumps in to control the glucogenesis. 3 different enzymes are necessary for glucogenesis, and they include glucose 6-phosphatase, pyruvate carboxylase, and fructose 1,6-biphosphatase. Aldolase is an enzyme that plays a part in converting glucose into energy. The most concentrated areas of aldolase in the body is found in muscle tissue. However, it is found throughout the entire body, but it's more concentrated in the muscles. When a person has muscle damage, the aldolase levels in the blood increase. Increased levels of aldolase can also signify liver damage and cardio-muscle damage due to things such as a heart attack. This particular enzyme plays a part in 6 reversible reactions during glycolysis and glycogenesis. There are different isoforms of aldolase. Glucose metabolism is made possible by the enzyme aldolase. It has 3 main isoforms, aldo A, aldo B, and aldo C. These three enzymes can be found in large quantities in the muscles, livers, and brain, as well as scattered throughout the entire body, but more concentrated in those other three areas, the muscle, liver, and brain. Different diseases and disorders cause a rise in the different forms depending on which one is being affected the most. These three isoforms of aldolase are located all over the body and have a functional part in glycolysis, fructolysis, and the creation of glyceraldehyde and ATP. And here you can see the different induction, modification, degradation. And you have your A, your B, and your C, and how those interact. And you have your RNA polymerase. Aldo-A is also known as fructase biphosphate. It's found in muscles and red blood cells. It is a glycolytic enzyme that plays a key role in glycolysis and glucogenesis. It is also said to be a scaffolding protein. This protein takes part in step 4, where the synthesis of the D-glyceraldehyde 3-phosphate and glycerin phosphate from the D-glucosebus is completed. And this just shows the structure, a molecular structure of aldo-A. Aldolase B has a role in fructose metabolism. Remember that fructose is found in fruits and in some vegetables. It can also be found in table sugar and other table sugar products such as corn syrup. Table sugar is also known as sucrose, which is a molecule made of glucose and fructose bonded together. As we eat food, these bonds are broken so that energy can be made. It is then used by all cells in our bodies during a process called metabolism. This part of the metabolic process takes place in the liver. The glucose molecule gives a phosphate group off the ATP it makes to a cell, which then allows the liver to use it. So remember, with ATP, phosphates are broken off, and that's what gives the energy. It makes the ADP, and then it goes back to ATP again. The first way it can use it is to make glycogen. The second thing it can do is go through glycolysis to make pyruvate. In a study by Wang and his cohorts, they found that aldo-B was downregulated in persons with hepatic cancers, which is the opposite of what aldo-C is known for. Hepatic cancer is one of the leading causes of death in Japan, so this study is helping to understand the role of enzymes such as aldo-B. Mutations in aldo-B can cause lower levels of aldolase B activity, which in turn cause different diseases and disorders. One hereditary disorder is caused by an autosomal recessive disease and is called hereditary fructose intolerance, or HFI. This disease results in an excessive amount of fructose 1-phosphate. This can cause several metabolic issues, causing liver and kidney damage. People with this disease will experience abdominal pain, vomiting, and hypoglycemia. It can be very lethal to babies who are in the process of weaning from breast milk. If a child is left undiagnosed, they could experience growth retardation, kidney dysfunction, liver failure, coma, and ultimately even death. A person with this disease should not ingest fructose. One study shows that 1 out of every 20,000 people are born with errors in metabolism that affects the digestion and breakdown of fructose, which I had no idea about. This is all very new and interesting to me that this occurs. The structure of aldo-B is very close to the aldo-A's structure. And when you look over here at the graphic, it just shows fructose being intook. It shows glycolysis and the phosphate pathway, and it shows the catalyst, aldo-B, occurring, and it just shows that portion in the intestines and the liver and how those are both affected. Okay, aldo-C is also an important enzyme in the 4th step of glycolysis. It is also involved in the reverse pathway of glucogenesis. This enzyme acts as a catalyst while converting fructose 1,6-biphosphate to, I can never say this right, glyceraldehyde, I think is how you say it. It is also a vital part of making ATP. While carrying on its main function, it is also involved in binding to the cytoskeleton of cells. It's been found to help out during hypoxia by participating in a stress response pathway for lung cells. This enzyme can be found mainly in the brain, more specifically in the hippocampus and Purkinje cells. It's actually been found that ALDO-C is involved in the non-glycotic positions as well as the progression of cancer. It is also highly expressed in several metastatic cancers such as lung cancer, gastrointestinal cancer, adenocarcinoma, and gallbladder cancer. However, it has been found that ALDO-C is an important factor for survival with glioblastoma patients. It helps to block the migration of cancer cells. This enzyme is also associated with areas of high metabolic activity. It can also act as a substitute for ALDO-B when metabolizing fructose. So if ALDO-B is not available, ALDO-C can actually come in and substitute for it. ALDO-A and ALDO-C are more closely related, however, than they are ALDO-B. So that's interesting too that can happen. This graphic shows normal cells along with ALDO-C as the enzyme and the process it takes and its results. And then it shows the NSCLC cells where the ALDO-C is free, and it goes down and shows exactly what is going on there as well. Aldolase B is an enzyme that fluctuates in activity due to dietary intake. The enzyme is part of the glycolytic pathway that deals with fluctuating messenger RNA levels in the liver. Aldolase B is controlled by several different elements, including the HNF1, HNF3, CEBP or EPB, and the DBP. A few of these are strong activators of the transcription process, while a few are weak activators. In this graphic, it just shows how some of these different factors regulate Aldolase B. So the first one is the hepatocyte nuclear factor 1, or HNF1. This is a protein that acts as a transcription factor to help control the activity of different genes. This protein is found throughout the body. However, it's more concentrated and needed in the liver and the pancreas. This protein is especially important for the creation and growth of beta cells in the pancreas. The pancreas is the major organ that produces insulin and beta cells. And beta cells are what produces and secretes the insulin. Insulin is needed to help control and regulate blood sugar levels in the body. The HNF1 factor also plays a part in liver development. It makes sense that it is also known as a tumor suppressor. This means that it helps ward off uncontrolled cell growth. Therefore, it helps suppress the onset of cancer. Protein interactions occur in the dimerization domain and helps to create a 2-protein unit called a transcription factor. Gene activity is also controlled by another region of the HNF1 called the DNA binding domain. This graphic shows the HNF factor, and it just shows the liver, the pancreas, the embryogenesis, organogenesis. The next one is transcription factor HNF3, or the hepatocyte nuclear factor 3. This is a protein that acts as a transcription factor. It includes 3 different proteins, the alpha, the beta, and the gamma. These factors work together in the expression of several liver genes. They're very similar to the carboxyl terminal on the amino acids in terminus. The HNF3 proteins function to regulate metabolism and differentiation of metabolic tissues. They are vital to the function of the liver and pancreas. These proteins bind to numerous genes involving gluconeogenic and glycolytic enzymes, serum proteins, and hormones. They are needed for activation of glucagon in the pancreas and activating enzymes to prevent the hypoglycemia. Here you have the condensed chromatin enhancer, the HNF3, where it's binding, and then you have the accessible enhancer. And so that just kind of shows the process that those also go through. The next one is the transcription factor, CEBPB. The CCAAT enhancer binding protein, or CEBPs, are considered promoters since they promote the expression of genes. These proteins are found in the nervous system and are in charge of making nerve cells. They play a huge part in learning, memory processes, and neural functions. They are found in hepatocytes, adipocytes, hematopoietic cells, the spleen, the kidney, and the brain. They function to aid in cellular responses such as proliferation, differentiation, growth, metabolism, and immunity. The CEBPs do not have an intron, but they include 4 exons. So, therefore, they have an alternative form for promoting and splicing. They are regulated by mechanisms such as phosphorylation and acetylation. These proteins have been found to be associated with fat development as well as osteoporosis. They have also been found to be useful as a tumor suppressor. And our last one is the D-box binding factor, or DBF. This is a protein that plays a huge role in the circadian rhythm. It's part of the PAR leucine zipper in most peripheral tissues. It plays a part in homeostatic aspects of sleep regulation. So it's also thought to be responsible or partially responsible for the synchronization between the hypothalamus and circadian oscillators. And we know that something as simple as sleep cycle and the other rhythms in the body it plays an important role in how our kidney functions, how our blood pressure, how we regulate temperature, regulation of our heart rate, our pulse, digestion with the peristalsis moving through the intestines, and even metabolism. So this actual factor is thought to play a huge role. And so a lot of our homeostatic imbalances or balances and maintaining homeostasis can be controlled with DBP. And so this is, to me, one of the, well, they're all important, but this seems to be a big one, a really big one, where if it's messed up and there's an imbalance, or if it's not working properly, it's going to actually affect everything else and how the body is ran. Thank you very much.